Guys, today we're going to be talking about the cell structure itself and the organelles that make it up. This is chapter 6 in the 9th edition of your textbook. That's the one that you guys have this year. And here's just kind of a really basic outline. These are the 3 big things I need you to take away from this lecture. And again, this is all the reading that you did for the summer work. And so this should all be really quick review, but you need to know 3 differences between prokaryotic and eukaryotic cells, the structure and function of organelles that are in both plants and animal cells, and then finally the structure and function of some organelles that are found only in plants or only in animal cells. So you need to know what makes them similar and what makes them unique. Okay? And just to give you an idea of kind of the size that we're working with here, this is an image from your textbook, and it shows you different sizes of things, right? So if you remember from chemistry, we have atoms way down here, 0.1 nanometer, right? And then we start to combine atoms and we start to make small molecules. And then those molecules might be getting bigger, or they might be made up of multiple molecules. So then we have lipids, proteins. We've talked about those with chapter 5 now. And up here we have ribosomes. So ribosomes are responsible for making proteins, and we're going to get to that. And then we have viruses. So viruses are a little bit smaller than bacteria. Okay, so bacteria is our first prokaryotic organism that we're going to talk about. And I just kind of want you to take a quick second to just appreciate the diversity of size that we're dealing with here. I don't really need you to worry about the different tools you may need to see things. So you can see here that from this point down until the end of this arrow here, these are the things that you can see with your own eyes. Okay, and then right here we have the point at which you can only see stuff with light microscopy or something better. Okay, and then here we have electron microscopy, so you can go even further down in size. You can see smaller things. Notice that we don't have on here anything that'll show us atoms directly or even small molecules directly. Okay, so I don't want you to get bogged down in that. I really just want you to think about these 2 things that are highlighted in yellow here. So in yellow, we have most bacteria, the nucleus of a cell, which we'll talk about, the mitochondrion of a cell, and then a little bit bigger than that, usually about 10 times bigger, are going to be most plant and animal cells. So we're going to talk about the differences between these bacterial cells and plant and animal cells, but for now, just realize that some cells are bigger than other cells. Okay? Now, to that, there are 2 types of cells that you need to be familiar with. We have prokaryotes, or prokaryotic cells, and they make up the domain bacteria and archaea, according to your textbook. Okay? And these are going to, pretty much for the purposes of our class, we're only worried about bacteria. Archaea are awesome, and there are some amazing organisms out there, but we really focus primarily on bacteria and then on eukaryotes. So this is the domain eukarya. And within eukarya, we have quite a bit of diversity. We have what we call protists. We'll talk about them later. They're kind of an interesting, not even really a true group. We have fungi. So mushrooms are the fruiting bodies of fungi. Yeast that you use when you make bread, that is fungus. Okay? Plants and animals. Okay, so there's a lot of diversity within eukarya and a lot of diversity within prokarya. Okay? Now, if you look here, we have a small 0.5 micrometer prokaryotic cell here. So this is our scale, right? And you can see here that the diameter is going this way, going width-wise here, is actually about the size of the scale. So I would say that this is about 0.5 micrometers wide. Okay, it's obviously much longer, and you could measure that, but if you compare that to the animal cells over here, here's just a single cell outlined in black, and it's way bigger. Okay. So, prokaryotic cells have some structures that are going to be pretty consistent throughout. I don't need you to memorize every single one of these. I really just want you to start to get an idea of the shape of a bacterium. Okay, so bacteria are often these kind of cylindrical shapes, not always, but often these cylindrical shapes. And they have these flagella, these like tails coming out of the back. Okay? They have a cell wall, which is something that you might have only thought that plants had from your middle school days or whatever. But in fact, there are cell walls in bacteria. It's a little different, but that's okay. There's a plasma membrane that's going to be a part of any cell. There are ribosomes. If you'll recall, ribosomes are the things that make proteins. So these guys need to do things, right? Any cell needs to do things. And so prokaryotes will also have ribosomes to make proteins because proteins are the things that do things, right? Now, just to highlight some of the differences, again, you do not need to memorize every single one of these differences, but some of the difference between prokaryotes and eukaryotes. Prokaryote literally means, if you were to translate it to English, it means before kernel. Okay, so if you think of corn or popcorn, you think of that weird thing in the middle, right? Especially with popcorn, you think of that weird hard thing in the middle that doesn't pop properly, right? Because it's the shell. It was the shell of the kernel. Okay? So the kernel is like the hard thing in the middle, and that's going to refer to the nucleus. Okay? So our nucleus is our kernel, and eukaryote translates to true kernel. Okay? So eu means true. That has a true kernel. So anything that is a prokaryote, that evolved before there was a kernel or before there was a nucleus. So all of these won't have a nucleus, whereas the eukaryotes have a true nucleus. Okay? Now, prokaryotes, because they don't have a nucleus, they have their DNA in a nucleoid, and I don't need you to memorize that. I just want you to realize that there's a difference here. Because eukaryotes, on the other hand, have the nucleus, and so they have their DNA inside of that nucleus. Okay? Now, cytosol is going to be consistent throughout both types of cells. Okay? That's just our nice little liquid medium inside of the cell. Prokaryotes don't have any organelles other than the ribosomes, whereas eukaryotes can have a huge diversity of organelles, and we have to talk about a lot of those throughout the course of the year, but really I just want this lecture to be a little bit of a primer on some of those organelles. Again, prokaryotes are typically smaller, and eukaryotes are typically larger. A nice average is 10 times as large as prokaryotes. Prokaryotes are considered primitive because they evolved much earlier on in evolutionary history, whereas eukaryotes are considered more complex. They have a lot more going on in terms of cellular mechanics. Got cut off a little bit there, but that's all right. So what I want you to start thinking about is cell size. So cell size is something that might seem simple at first, like, oh, some cells are big and some cells are small. But here's where it gets a little tricky. Cells have to have a particular, what we call a surface area to volume ratio. So you've heard of volume, you've heard of surface area, right? And it's just a mathematical representation of those two things. Okay? So if you have a large surface area, that allows you to interact with your environment more. Okay? And cells need to interact with their environment all the time. So if you think about it, if you think of your whole body as one big cell, obviously it's not one big cell, but just think of your body as one big cell. How are you going to interact with the world around you? Well, if you think of your skin, that's kind of like a cell membrane. Okay? It's this big cell membrane. So that's really how I interact with the world. So the more cell membrane I have that's able to access the air or the water, whatever I'm in, that means the more I'm going to be able to interact with my environment. Okay? And cells are the exact same way. They need to have a large surface area to volume ratio in order to effectively put stuff in, or put stuff out or take stuff into their cells. Okay? Now, here's where it gets a little tricky and you need to start visualizing things. So here, I want you to picture just a small little cube. This is from your textbook. But a little small cube here. And this cube has a surface area, right? If you took the area of every single side, there are 6 sides of a cube. Well, if I took the surface area, or just the area of one side, and I multiplied it times 6, because they're all the same, because it's a cube, okay, then I'm going to get some surface area. Okay, so let's say here it's going to be 6, okay, 6 units squared, whatever it is. Doesn't matter right now. Okay. Now my volume, I have to take my height, my width, and my length. Okay. My volume is just going to be 1 times 1 times 1 in this case. Okay, because each side is a unit of 1. Whatever that unit is, it doesn't matter right now. Okay. So in order to find my surface area to volume ratio, I literally just have to take my surface area and divide it by my volume. So 6 divided by 1 is 6. Okay, so that's a small cube. Let's look at a big cube. Okay, so this cube is 5 times as big. Okay, and that means that the length of each side is going to be 5. And so now if I want to find the surface area, I have to find the area of each side. So I'm going to do 5 times 5. Okay, that's going to give me, let me get my pen here. That's going to give me 25 units squared here. But I have 6 of those sides, right? So each of these is going to have 25. And so I have to do the 3 other ones that aren't shown either. And so if I add all of those up, I'm going to get 150. Great, that's just really simple math. But if I do total volume, I have to take the height, okay? I have to take the width, and I have to take the length. And so when I do 5 times 5 times 5, I'm going to get 125. Now I need to calculate my surface area to volume ratio again. And if I just do simple math of 150 divided by 125, I'm going to get 1.2. Okay, so what you might be noticing is that my thing got bigger, right? It went from a tiny little cube to a big cube. The ratios were the same, right? It's still a cube. Each side is the same length. However, because it's bigger, my surface area to volume ratio is actually way smaller, 6 compared to 1.2. Okay? So instead of doing a lot of big cells, what organisms typically have is a bunch of small cells. Okay, so here is the same size thing, but made up of all little cubes. Okay, so here we can increase our surface area and keep our same volume, and therefore we can increase our surface area to volume ratio. Okay, so I can get back to that 6 by just making up the same shape out of a bunch of little shapes. And this is going to be really hard to visualize sometimes, so I really recommend that you draw some different shapes of different sizes, you assign lengths and widths, and you do some calculations yourself. Okay? But this really matters for cells. So here's an example of a situation in which you really need a high surface area to volume ratio. So this is the inside of an intestine, okay? And we have these villi on here. Okay, these villi actually have microvilli, and if you look here, the villi are like these little extensions. So it's all these like little, almost comb things coming off of the edge of the intestines, right? And so here's a cross section of that. So you can see that there's some blood capillaries, and there's some little cells here, right? But they're little projections, right? They're little like tentacles almost. Well, these tentacles, if I zoom in on some of these epithelial cells, okay, so I'm going to zoom in right here. Those have their own little tiny projections. They have their own little villi. So we have villi, and then we have microvilli on top of the villi. So here we have tentacles on tentacles. Okay? And the whole reason we do this is because, well, your intestines are involved in a lot of nutrient absorption. Okay? So you need to eat stuff, and then you digest stuff, and the intestines are the last stop for digestion. Okay? And during that process of going through the intestines, we're taking in a lot of those nutrients. In order to take stuff in, I have to interact with my environment, right? In order to interact with my environment, I need to bring stuff in. How do I bring stuff in? Through the surface. Okay, so I need a high surface area here. Okay? So that's why our intestines are so highly folded. Okay, the texture of the inside of a small intestine has typically been described as being like velvet because it's just so many of those little projections. It feels like chinchilla fur, right? Like just so soft. Okay. Now, I want to talk a little bit about plants and how they differ. So plants also need surface area, but you're going to see a lot of comparisons to their root hairs. So they have roots, right? That root is just going straight down on the ground. But then off of the root, we have these little root hairs that go out. And you'll notice that they're very thin, just like hairs. And the reason for that is because if you have something that's really long and thin, that's going to have a lot of surface area. So anytime you see, oh, I have a lot of surface area needed here, you're going to notice long, thin projections, just like those villi. Now with that, I kind of want to talk about just a few of the organelles that you guys have learned about. This is not necessarily going to be a comprehensive list because you do need to read the chapter if you haven't, which you better have because it was summer work, but this is really important. Okay? Now, again, organelles are a thing that are pretty much unique to eukaryotic cells. prokaryotic cells only had the ribosomes, right? And what's cool about organelles is that they are like their own little space. Okay, so here I have an animal cell on the left and a plant cell on the right. And you'll notice that all of my organelles are labeled. So I have ribosomes, I have a lysosome, I have a centriole, I have a centrosome, peroxisome, etc. Okay, often these are like little bubbles. Okay, you see all these little bubbly structures? So they're like their own little compartments inside of the cell. And that lets the cell kind of keep stuff where it belongs, right? So if you think about your house can be like a cell. Now, if you didn't have any walls in your house to separate it into rooms, okay, you just had one big room, basically. Instead of having separate rooms, you had one big room. Well, you might get really annoyed by your parents or your siblings or anyone else at home because they're always doing something in the same room as you. But if you have your own little space, like maybe the lysosome has to do its own little thing inside of here. And so I don't have to do it along with the DNA inside of the nucleus, right? I have my own special little space. It's the same size house, but I get my own room. Okay? And that's really important because some of these organelles are involved in some really destructive stuff. So like the lysosome, for example, is responsible for breaking some stuff down. Okay? And so you don't want it to accidentally break down things that are good, right? So you need to keep stuff in their place. Ribosomes, these are our basic protein makers. These are our protein factories. Okay, for now, I just kind of want you to picture it as two little pieces here. We have two different subunits. Notice that they are slightly different in prokaryotes as they are in eukaryotes, but it's really the same idea. Okay, basically, these are going to make your proteins. That's all you need to know for now, because we're going to get more and more into that when we start talking about gene expression. Okay. The inside of a ribosome looks like this, and if you look closely, this is kind of taking us back to our proteins. If you look closely, you have amino acids being chained together to form a protein. Okay. The endoplasmic reticulum is outside of the nucleus, okay, and there are two different types. There's the rough endoplasmic reticulum. You can usually abbreviate this as RER. And this is going to be all about protein synthesis. It's called rough because it has all these ribosomes on it, so it kind of like is all bumpy. Okay? But you can also just remember that R stands for rough and for ribosomes. Okay? However helps you to memorize it. But all these ribosomes are what we just saw. Okay? So what I'm doing is I'm just zooming in on a single one of these. Okay? And this is a cross section of it. This is what's happening inside of it. Okay? Then there's also, I thought we were going to go on to smooth ER, but, okay, here's the smooth ER. So the smooth ER doesn't have those ribosomes. So here you'll notice, oh look, I have all of these ribosomes all over here. But this area doesn't have those ribosomes. That's why it's called smooth, okay? And it's going to be dealing with lipids. It's going to be dealing with fats. Okay, generally, it's going to be involved with making fats, and we'll talk more about that later. But we're going to move on to the Golgi complex. Okay, so this is a membrane bound, so that means it's encased in a membrane. Okay, that consists of some sacs, and I don't need you to memorize this term of cisternae. Okay, you do not need to memorize that. But just know that the Golgi complex is going to be about transporting stuff, kind of packaging stuff and transporting it. Okay. You're going to synthesize and package stuff, and then you're going to move it throughout the cell or even out of the cell. But it also makes the lysosomes. Okay? So this organelle actually makes other organelles. Okay? So just kind of zoom in and explain what a vesicle is, because that's going to be really important. A vesicle is basically a little bubble. And it's just a little bubble inside of a cell that can hold stuff. So if you remember, if I go way back to this picture right here, I like to have things in their own place. Okay? Now, if I am transporting something dangerous from, let's say, this part of the cell, and I'm trying to get it out of the cell, maybe it's a poison, something like that, I don't want it touching stuff, right? So I'm going to put it in its own little safety bubble and then let it leave. And we're going to get more into that with the next chapter. But just realize that we need ways to transport things. Okay? And so that's what vesicles are for. They're little bubbles that you can transport stuff in. Okay? Excuse me. Now, lysosomes, so this is the other thing that is made by the Golgi complex or the Golgi apparatus. Okay? Lysosomes are these like little sacs, and they are going to contain hydrolytic enzymes. Okay? That sounds really fancy. All you need to know about it is that those are going to be used for digestion of things. So I don't want you to think about digestion like it happens in your body, but it's really kind of the same idea. It's just breaking stuff down. Okay? So there's stuff inside of here that will eat stuff away. And so if you need to break something down, you're going to send in your purple lysosome here. Obviously, it's not necessarily purple, but you're going to send it into this vacuole. Okay, so I have a vacuole here. It has some food. And, ooh, look, food. I need to digest that food. So what I'm going to do is I'm going to combine these two vesicles. This is really kind of just like a vesicle. And now that they're in the same place, I can break stuff down. Okay? Now, this is also going to be how a cell kills itself. So sometimes cells have to kill themselves in order to be better for the organism as a whole. We call that apoptosis. Okay, and we're going to talk about that more when we talk about cell cycle. Now, the mitochondria, you probably know it as the powerhouse of the cell. It's way more interesting than that, and it's a shame that that became such a thing, but here we are. So the mitochondria has this really classic shape, often shown in, well, it really depends on the type of organism. You often see it in green, which is kind of surprising because usually chloroplasts are green, but sometimes you'll see it in this kind of pale peach almost color. Okay? So there are multiple membranes here. There are 2 membranes, and that's really, really important. And I don't want to get too bogged down in why this looks so complicated and what's happening inside, because we're going to get to that when we talk about cellular respiration. So for now, just know that the mitochondria is like a factory for energy. Okay. Going to skip through this because I want to talk more about it later. Okay, vacuoles. So vacuoles are basically all about holding on to stuff. Sometimes they're going to help with digestion, right? And sometimes they're just going to hold stuff. Okay? They're going to hold stuff until we can get rid of it. Plants are a little interesting because they have vacuoles, just like animal cells, but their vacuoles can be huge. So here, the blue thing here is a giant vacuole. Okay? And it's filled with water and a bunch of other stuff. And this being so full actually allows the plant to be pretty rigid. So this is so full of liquid that it's actually going to push against the cell wall and kind of force it to be nice and rigid. And that'll give a plant the ability to fight gravity, for instance, okay, along with some other things, but we'll talk about that later. Chloroplasts, speaking of plants, are only going to be in our plants and algae, things like that. Algae is misspelled up here. Whoopsie. Now, these are going to be responsible for photosynthesis, and we have an entire chapter dedicated to photosynthesis. So I really am just going to show you what they kind of look like. Okay, so this whole thing here is a plant cell. Okay, notice it has a big central vacuole here. And then here we have a chloroplast. Okay, so the chloroplast is this green organelle with a bunch of these little, they look like pancake stacks. Okay, we're going to talk about that to death in photosynthesis. Okay. They contain chlorophyll. You've surely heard of chlorophyll. It's the thing that makes plants green. Okay. For now, I don't need you to memorize differences between chlorophyll A and any other kind of chlorophyll. Okay. Just think of chlorophyll as being green for now. Okay. This is what that chloroplast looks like. Okay. So it also has 2 membranes, just like the mitochondrion does. So we have the outer membrane, we have the inner membrane, and then inside of that we have all these little stacks. Okay, and there's also this nice fluid inside. Each of those pieces of a stack is called a thylakoid. Okay, I just kind of want you to get familiar with this structure and be able to diagram it. So I would practice diagramming this, just really basic. Okay, you don't even really have to show them connected like this. You can just have stacks of pancakes inside of here. Make sure you get the 2 membranes, and be able to tell me that that's only found in plant cells or algae cells, things like that. Okay. Now, those stacks, okay, those thylakoids, remember those are the pancakes, those are stacked into a big stack called grana. Okay, so this is 1 granum. chlorograna, made up of 3 thylakoids. Okay, so each of these is the thylakoid making up the grana. And this is where all of the light reactions happen during photosynthesis. So you have light coming in, and then a bunch of stuff happens in here, and it's really, really important, and this is really important, and this is really important, and this is really important. That's great. We will talk about it. We will cover it. You will learn to hate it and love it at the same time because it's super interesting, but it's super complex. Okay? But for now, just know that the chloroplasts are all about light. Okay? Here's kind of a nice little overview of the organelles, just to kind of give you a nice, pretty extensive look at how busy these cells can be. Okay? Cells are very busy places. A lot of things are getting done, and a lot of things are doing those things. Okay, so just kind of get familiar with having a very stressful looking diagram and being able to figure out what each piece might be doing and what each piece might look like. Okay? If you're still struggling, here are a couple of extra videos that I would recommend watching. I would just Google the phrase there instead of typing out the URL. But that's going to do it for organelles, and next time we're going to be talking about cell membranes.